Hello, my name is Suman Rice, and I'm chair of the Society for Reproduction and Fertility. I'm delighted to welcome you to the SRF 75th Anniversary Lecture Series. This collection of talks celebrates 75 years of the Society, its rich history, and its ongoing contributions to reproductive science. The Society was founded in 1950 as the Society for the Study of Fertility, bringing together a remarkable mix of scientists and clinicians, from reproductive physiologists to endocrinologists, pioneers of livestock breeding, and experts in human fertility. Each lecture in this series highlights the diversity of research and innovation in our field, reflecting the interdisciplinary spirit that has defined the Society from the very beginning. We hope these talks give you a sense of the Society's achievements, the work of its members, and the dynamic community that continues to advance reproductive science.
So it's a great pleasure to be able to deliver a lecture for the SRF 75th Anniversary Series. The Society has been very important to me during my career, and I've been a member since 1983. And today I'm going to speak about new approaches for fertility preservation, specifically on lab-grown eggs. So if we start with the formation of the oocyte, so the primordial germ cells that migrate to the gonadal ridge and then form nests surrounded by somatic cells, and at that point they will start to enter meiosis. And then the nests break down and each individual gamete will be surrounded by somatic cells, and at that point they're arrested at the diplotin stage of prophase 1 of meiosis. And this forms the fundamental developmental unit of the mammalian ovary, the primordial follicle. And this is a pool of germ cells that must last the female throughout her reproductive lifespan. So the follicles, once they're formed, they will be activated. Some of them will be activated to grow. And this activation process is something that's continuous throughout life until menopause in humans. And once the follicle is activated to grow, the oocyte develops, the somatic cells proliferate to form multilaminar preantral follicles, and then the follicles form a fluid-filled cavity, the antrum, and at that point the cells around the oocyte become the cumulus granulosa cells in preparation for ovulation. And in humans, it's normally only one of these will be ovulated per cycle. So as we know then, in most mammalian species, it seems that there is a fixed number of these eggs and these decline with age. So you can see in the blue bar that they drop in numbers of eggs as we move towards menopause. But this rate of loss can be accelerated, and that's the purple line. And that's accelerated if the woman is undergoing some treatments. So, for example, chemotherapy and radiotherapy, can be a dramatic loss of germ cells, rendering the woman infertile. Now, these immature eggs, of course, throughout life, 99.9% of them will degenerate, and only about 0.1% of them will ever be ovulated. So the question is, can we save these eggs and preserve fertility by culturing immature eggs to maturity? So what can women do to preserve or restore their fertility? What are their options? And this is really just, looking at how the whole field of fertility preservation has progressed in recent times. So we know that prior to 1993 and the advent of egg freezing and embryo freezing, women undergoing these treatments were offered the opportunity to either freeze eggs or embryos. But that's often not an option because of the hormonal treatment, and also it can cause a delay to the treatment. And this was the only options that women had prior to 1993. And then in 1993, there was a new development, and that was the ability to freeze ovarian cortex. So to take pieces of the ovarian cortex that contains these primordial follicles, to freeze that, and then perhaps to transplant it. And that was an option that became available to prepubertal girls. And indeed, it's the only option currently available for fertility preservation in prepubertal girls. And this new era in fertility preservation was really brought in by Roger Gosden. Roger Gosden was my PhD supervisor in Edinburgh, and then he went to Leeds and then to the United States, and is currently in Virginia in the United States. Roger's the one on the right of the photograph, or my right. Roger's early work in Edinburgh was central to developing ovarian tissue cryopreservation and ovarian tissue transplantation. And really, this work transformed the field of fertility preservation. And Roger collaborated with other scientists and clinicians within Edinburgh. He collaborated with David Baird and Bob Webb, and these are Bob's a scientist and David's a clinician scientist. Both of them were Marshall medalists of the society, so well known within the society. And sadly, David is no longer with us. But they did some really critical experiments with Roger, where they took pieces of the sheep ovary and cryopreserved it. And the sheep were infertile, and then they transplanted the frozen thawed tissue. And in the first series of experiments, they were able to produce a lamb. And this is Frosty, the lamb that was born from the cryopreserved transplanted tissue. So that really gave proof of principle that transplantation of these cryopreserved ovarian cortex pieces could result in healthy eggs and a viable pregnancy and offspring. And what that led to was the translation very, very quickly into a clinical service. The successful experiments with sheep, it showed that it could be a clinical service. And in 1993, the first young woman had ovarian tissue removed and cryopreserved using the protocols that was developed by Roger in mouse and subsequently sheep. So it's a really nice example of translation of basic science into clinical science. And so now ovarian biopsies can be taken and cryopreserved. And these pieces of tissue, they contain the most immature eggs, and it's the only option, as I've previously said, for prepubertal girls. So you can see the pieces of tissue here that would be cryopreserved. And this tissue contains or may contain hundreds or thousands of immature eggs. That's variable in terms of how many they would contain. And then when these pieces are transplanted, and this has been done all over the world now, and the first centre was Jacques Donné's in Brussels to achieve a live birth, but there's now been over 200 live births using this technique. But transplantation is not suitable for everyone. So, for example, women with blood-borne cancers, you can't really take the risk of transplanting the tissue because it may contain malignant cells and you would not want to reintroduce the cancer. So what are the options for that tissue, all of this tissue that's stored, particularly for prepubertal girls who this is their only option for fertility and that frozen tissue? And that's where we've been working on lab-grown eggs. So trying to grow that tissue outside the body to the point where we can obtain oocytes, eggs, that will be capable of being fertilized and forming embryos. So this method is, I refer to as in vitro growth, IVG, the development of immature oocytes to maturity entirely in vitro, so growing the eggs that are already there, the old eggs, and the in vitro gametogenesis or in vitro derivation is the formation of oocytes from stem cells. And I'm not going to talk about that in detail. I'm going to focus on the lab-grown eggs and then mention the in vitro gametogenesis or in vitro derivation. So lab-grown eggs have many applications. They're really essential for in a human system for basic science, because we really need to understand more about the human ovary and how various factors affect the human ovary. So a culture system allows us to tackle many questions within the human system. It's also important for fertility preservation in cancer patients. And of course, for in vitro fertilization, it could be considered the in vitro fertilization Mark II, a new version. In domestic species, for cows and sheep and other species, it could have a big impact in terms of shortening generation intervals, and of course for the conservation of rare breeds. So there are many reasons to develop lab-grown eggs. So I'll just give a short history of developing eggs or oocytes from mice in vitro, because of course mouse was the first model to be used to be able to develop systems to grow oocytes outside the body. And this has gone back to the 1930s. So 1935, groups were looking at taking the oocyte and its cumulus mass and trying to mature it, meiotically mature it and fertilize it. But it wasn't until 1984 that that was achieved, that you could mature them, fertilize them, and then develop them, obtain live young. And then there was moving it back in the developmental phase, starting not with antral follicles, but with pre-antral follicles, so more immature. And this started in 1979, developing systems to grow these eggs outside the body. And it was in 1989 that was able to mature them, fertilize them, and obtain live young. So it's taken quite some time to develop these systems, even in mouse. And going back even further into the primordial stage, where John Eppig's lab was able to grow these entirely in vitro and obtain live young. Now, if I just take you back to the 1980s, because then if we went to meetings of SRF or SSF, we would find that not many people were talking about culturing pre-antral follicles. In fact, most of the work was involved with the later stages of development, not the earlier stages of development. And worldwide, there were only 3 groups working on this. There was Gilbert Greenwald, there was John Eppig, and there was Roger Gosden, us in Edinburgh. And it took a while, but there's now hundreds of groups working in this area. So if we go to meetings now, we'll see lots of posters and hear lots of presentations in this area. And just to remind you that the first mouse that was born from this technique was Egbert. And Egbert was only one in that particular experiment. And Egbert was not an entirely normal mouse. So there's been modifications, improvements to the mouse system. And it's now been shown that the offspring that are now born are more in keeping with the in vivo grown oocytes and embryos. So there's been improvements, big improvements on the mouse. But now the challenge is to develop these systems for humans. And we've been working on human ovarian biopsies that contain mainly primordial follicles, the immature eggs. And that's our challenge, is to develop them to maturation and fertilization. So I'm going to take you through developing human oocytes from the primordial stages entirely in vitro. So firstly, we have to optimize the growth from primordial stages. So the optimization of activation of primordial follicles, and then support the development of isolated growing follicles, so maintaining the oocyte somatic cell interactions and growth and development of these pre-antral follicles. And then the final stages of oocyte development, the oocyte granulosa complexes, to obtain mature oocytes that will resume meiosis and hopefully can be fertilized and produce normal embryos. So this is our starting material. This is the pieces of ovarian material that we have, and we prepare it so that we can get activation. And the key to getting activation is the preparation of the tissue and the architecture of the tissue, because this interferes with a lot of the signaling pathways and activates the follicles to start to grow. And once they start to grow and they reach pre-antral stage, we can then remove them with some of their theca cells, and we can isolate the individual follicles and grow them individually. So many people have been working on activating primordial follicles from the 1990s in Outi Hovata, and you can see here many people are working on them. They're all culturing for different times, so that's very variable, the length of time that's required to get them to a particular stage, and it depends very much on the preparation of the tissue and the additives. So we dissect them out, we don't use enzymes, and then we grow them individually in V-shaped wells, and we do not embed them in a matrix. We add activin and ascorbic acid and low-dose FSH, and then we can get them to grow further to the antral stage. And this shows you the antral stage of development. It shows you the pre-antral follicle that still has nice oocyte communication, oocyte somatic cell communication, and that's dependent on activin being present. This shows you a nice antral follicle, and then what we do is we remove the oocyte. We don't want really large follicles. We want to try and develop this oocyte with its surrounding cells and get it to a point where we can mature it. There's been several groups that have been working on this in human, but much less than from the earlier stage. And again, the groups, these highlighted are our group, but the other groups, it's a range of times that they've been culturing them for, from as low as 5 days up to 42 days. So again, incredible variation. And then we've matured these oocytes. We've looked at in vitro maturation, and we've obtained metaphase 2 stage oocytes, albeit with large polar bodies. But it's a proof of principle that it can be obtained. And this just is a summary of our in vitro growth of human primordial follicles from this strip up to maturation. And again, just to highlight the other groups that are working on this, much less. There's only 3 groups that have so far achieved this, starting from, well, actually only 2 with primordial follicles. The first group was really from the isolated follicles that had already started to grow. Again, a range of times, and one group used alginate and the other 2 not. So this is our group here, and again it shows that this is a difficult process. So there's fewer people working and taking them through all the way from the earliest stages. So, but we have developed a multi-step culture system that supports human oocyte development. So we can take a piece of tissue from human ovary, we can activate growth, we can isolate growing follicles, and we can get antral formation, we can remove the oocyte and its surrounding cells, and we can get them to reach metaphase 2. So we have losses, of course, at each stage of culture, and that's why we need to optimize each of these stages, and that's ongoing work. I'd just like to highlight that there's a group in China that's been able to grow human secondary follicles, and they've been able to get metaphase 2 oocytes, they've been able to fertilize, and they've obtained blastocyst formation. So this is a really good proof of principle that in vitro grown oocytes could indeed form viable, competent oocytes that will form be fertilised and form embryos. So we're working on improving our culture conditions. We focused on improving our polar bodies, and we've changed some of the physical conditions, particularly oxygen tension. And we can see that in some of the oocytes, we don't see the large polar bodies. We look at different tissue sources, and healthy women at the time of caesarean section, we get the best oocytes from them that we can mature to metaphase 2. Prepubertal girls' tissue is rather different because the tissue, depending on the age, grows at a different rate. So it's slow growing if it's from 3 to 10, and slower, faster growing, but not the same as the adult tissue between 12 and 15. So that means that we have to adapt the culture system for the prepubertal and young tissue. Turner's patient, where a lot of the primordial follicles are really degenerate. We have been able to culture up to the early antral stage, but it's very few that we're getting, and they're almost mature, but we're not quite there yet with the Turner's tissue. And then there's a gender reassignment, chemotherapy-treated patients, and that's really dependent on the chemotherapy that's being used. Some we have normal oocytes, and others we just see degenerate oocytes after growth. So it's very much dependent that you have to, one size doesn't fit all, and we have to adapt our tissue type to the patient group. So that's ongoing work. So for us then, the next steps are determining the health and developmental competence of the in vitro grown oocytes, and doing sequencing of epigenome and the metabolome. We've applied for HFEA approval to be able to fertilise these in vitro grown oocytes. So watch this space, and we're hoping that that will be approved and that it will happen relatively soon. And then, of course, we've been positioned to test embryos, but we also have parallel studies going on large animals in sheep and cow, and we're doing things like embryo testing and transfer of live young, particularly in the sheep, and that's with Helen Picton in Leeds and Colin Duncan in Edinburgh. So you'll hear more about those exciting results from those models soon. So I've spoken about the multi-step culture system that we can use in humans, and we studied the effect of age, chemotherapy, and the role of signalling pathways. But we're now using it to look at supporting new oocyte formation from stem cells. So this is just some of our publications and the other systems that we've approached using the human culture system. And what we've been looking at is making gametes from stem cells. And this is an area both in making oocytes from stem cells and sperm from stem cells. It's an area that many, many groups are interested in. So, of course, the first phase is to differentiate the stem cells to germline cells and then combine them with somatic cells, and in the case of the oocyte, to form primordial follicles, and that's in vitro derivation. But really, we need to improve on this phase 2 of our in vitro growth and combining it with cells from the ovarian tissue to allow these follicles to start to grow fully in vitro, and then we can go through the process of in vitro maturation and in vitro fertilization. So again, this is ongoing studies. So in summary then, the future of fertility preservation, discovery science is reshaping. And we need to think now about protecting the ovary from these damaging therapies. And there's lots of nice work from my colleague in Edinburgh, Rosanne Rosario, where she's looking at AMH and protecting the ovary from the damaging effects of various chemotherapeutic agents. And that is ongoing, and she has very positive results in that. Also preserving the ovary. So, of course, the preservation of ovarian and on the male side, testicular tissue. Very nice work going on from Rod Mitchell in Edinburgh, and hoping to do testicular transplantations for tissue that's been cryopreserved. and of course it's ongoing in the ovary, but with the ovary also looking at propagating. So in vitro gametogenesis, so making new cells, new oocytes, or using the primordial follicles. And of course, preventing some of the effects of the aging process, or the abnormal processes that happen. And that is really requires a greater understanding of the cell processes within the human ovary, and the in vitro models will allow us to target those. So protect, preserve, propagate, prevent strategies have the potential to extend what is possible in fertility care. And I would direct you to our review in Physiological Reviews. So I'd like to thank all the people who have been involved in this work and the funding bodies, and also acknowledge that the Society for Reproduction and Fertility have funded many summer students in our lab, and we thank them. But finally, I would like to say the importance of the Society to me throughout my career. Because as a PhD student in 1983 and joining the Society, I've always been inspired at meetings. So the Society and its journal Reproduction have been very important. And not only that, I've been incredibly fortunate to meet trailblazers and role models. And I'd like to highlight 3 of these role models that are sadly no longer with us, but whom I met as a young student at these meetings. And Barbara Weir, Anne Greta Biscoff, Anne McLaren, who are not only inspiring women, great scientists, and have a legacy, a great legacy within the Society. So thank you very much for your attention.